Welcome to Structure Club. I'm Scott Stagg. I'm a professor at Florida State University, and I use cryo-EM to determine the structures and mechanisms of proteins involved in vesicle trafficking.
And I am Ashwant Francis, an assistant professor at Florida State University. My lab focuses on structural biology of viruses and how they interact with host cells. Together, Scott and I created Structure Club as a journal club podcast and YouTube channel where the papers are given by the authors themselves. Today, speakers are Mike Cianfrocco and Somaye Badieyan from the University of Michigan.
Welcome, Mike and
Hello.
Somaye.
Hi, hello. Glad to be here.
Thank you so much for inviting us.
Nice to have you guys. Mike is an associate professor at the University of Michigan, and Somaye is a research investigator in his lab. They will be talking with us today about their recently published paper in Science Advances. It's called HIV-1 binds dynein directly to hijack microtubule transport machinery. Mike and Somaye, y'all are going to be presenting the paper together. I think Mike is going to start us off. So Mike, take it away.
Sure. So, thanks for having us. So we'll take you through the paper today. Samaya, you can pull up the slides that you have for us to go through. While that's happening, the backstory here is that as I started my lab in 2017, people had just linked the dynein machinery more specifically to HIV trafficking. And that's also when Samaya joined my lab at the same time, she was looking for a project to come in and take over and start a new area of the lab. And so for us, we really wanted to understand how dynein was connecting to HIV at a molecular level. And I just come off a postdoc where I became an expert in dynein regulation. And so it felt like we should be able to tackle this problem. Like many things in science, it's much harder than you think it is, but we were excited, and Samaya was also excited. So here, Samaya, you can sort of take off.
Sure. Thank you, Mike. And again, thank you for inviting me to this podcast. So as you can see, the title is HIV-1 binds dynein directly to hijack microtubule transport machinery. But actually, it was a very surprising outcome of this study, and we didn't really know at the beginning. What we knew, okay, do you see my slides moving? Okay,
Yeah.
great. So, but first, let's talk a little bit about the life cycle of HIV-1. HIV-1 replication starts with the fusion of its enveloped to the membrane of the host cell, where the whole capsid, the whole core, that means the HIV capsid plus all the other component of the HIV-1, including reverse transcription and the other components needed for the further replication, as a whole will be released to the cytoplasm. Due to the dense cytoplasm, the dense environment of the cytoplasm, HIV-1, like many other viruses, depends on the microtubule machinery, and it has, during the evolution, it has evolved tools to hijack microtubule machinery to move from the cell periphery to the nucleus when the rest of replication happens in the nucleus. So from the retrograde transport of HIV-1 has been shown to be dependent on the motor protein dynein. As you can see it in this picture, it has been shown in the light purple over there. But dynein, for its motility on the microtubule, it is always toward the minus, exclusively toward the minus end. It depends on its activator, dynactin, about 2.4 megadalton structure, and another class of the cold protein, dimeric cold protein. That over here I showed those the dimeric protein, or known as dynein activating adapters. The N terminus is interacting with the dynein and dynactin, while the C terminus is available and specialized for interaction with the cargos. Over here, the cargo we have is HIV-1. So from this cold activating adapter of the dynein are huge growing class in a really weekly basis, you will see in the publication that the new adapter has been found for the dynein. But from this growing class, BICT2 has been found to be the cargo adapter of the dynein for HIV-1. There were several publications on that. So to study the mechanism of interaction, we set to reconstitute biochemically out of the cell in vitro this movement of HIV-1 in presence of the dynein, dynactin, and of course full-length BICT2. Everything at the beginning seems to be easy. We needed the only thing that we needed is the purified core, because as I said, the core stayed fully intact inside the cytoplasm till it reached to the capsid, so to the nucleus. So we needed just the core of HIV-1 for this experiment. And so our collaborators from the Vanderbilt University provided HIV core, which was fluorescent. It means a component of that VPR was fused to GFP. So we were able to monitor them during the single molecule assay. For this single molecule assay, we immobilized microtubules on the cover slip, glass slide, and we were able to monitor the movement of the fluorescently labeled cargo in addition to any other fluorescent component, for example, dynein or activators on the cover slip. So we started these experiments. The first thing that we noticed over that, we found that we weren't able to see any motile events of HIV cores in presence of the dynein, dynactin, and BICT2. That was actually a bit unexpected and took me about 2 years to eventually come to a state to find how they can move and what's the reason that they are not moving. And these 2 years, my experiment, my single molecule assay was kind of like a pot, really. I was trying to add anything that may play a role in this movement, especially the host factor interacting with the capsid. I used to add to this combination to see if eventually I can make this work or not, but it didn't. So it made me to think about, after all, if BICT2, because BICT2 is a cargo adapter, is at all interacting with the capsid or not. So I ran a setup of drop-off experiment, and those drop-off experiment was super surprising, because I found that, okay, if I don't have any BICT2, dynein, and dynactin, and over here in this picture, you see microtubule in magenta and the HIV-1 as these green dots, I don't see really that many HIV-1 on the microtubules. But when I'm having, as already showed to you, when I'm having BICT2, dynein, and dynactin, I can see many recruitment of the HIV particles on the microtubule. To my surprise, when I excluded BICT2 from this mixture in the single molecule assay, the number of recruitment were not affected at all. I removed the dynactin, again, nothing. But when I excluded dynein, the number of HIV-1 recruited to the microtubule significantly reduced. And eventually I noticed that the only thing I need to recruit dynein, to recruit HIV-1 to the microtubule, is only dynein, and the BICT2 is not needed for this case. And over here, co-localization also showed that on the microtubule, HIV-1 can be localized with the dynein molecules. The next thing, I think, is another surprise to me, was that when I used other activators, activating adapters of the dynein other than BICT2, because BICT2 is in auto-inhibited state, and the presence of cargo and its interaction to the cargo binding dynein of the cargo dynein binding domain of the BICT2 is needed to activate BICT2. So the BICT2 as a whole, the N-terminus of that, can interact with the dynein and dynactin to generate motile events. And I hypothesized, if the dynein by itself is needed for interaction with the HIV-1 and to bring HIV-1 to the microtubule, so it means that it's not able to activate BICT2. And if instead of BICT2, I use another activating adapter that are constitutively active, it's not in auto-inhibited state, I should be able to generate motile events. And it was right. At the end, as you can see with the BICT2, I wasn't able to see in the motile, in the single molecule assay, I wasn't able to see any activity. But when I replaced the BICT2 with the truncated BICT2, just the intermediate, so it is not anymore auto-inhibited, I was able to see some motile events. And the same for the BICT2, for the same as BICT2 N-terminus truncated, I switched to BICT-R1 as a full length or BICT-R1 truncated. Both of them, both BICT-R1 as a full length also is constitutively active. And the same as the N-terminus one. For both of them, I was able to see motile events. Both BICT2 and BICT-R1 belongs to the same group, and so I just switched to totally different groups, classes of this activating adapter. Hook3 and dynein, they are totally different, and both of them, they were truncated version. There was no the proposed cargo binding domain were deleted in both of them and just work on the N-terminus part. And with in both cases, with the Hook3 and dynein, I was able to see motile events. So again, it is proof that because binding happens directly, anything that can activate the HIV1, the dynein that is bound to HIV1, generate motile events of HIV, as you can see over here as well.
So can I just can you go back one? So these are called chymographs, right? That's the right word?
Yeah.
So one thing that I think might be useful for a listener is just briefly explaining how these work, because I've seen that, speaking from my perspective, the first time I saw them, I'm like, I don't understand how to interpret this. I see that they're different, but but it's such a cool technique, and so I think it's probably worth just like a minute to sort of
Sure,
help.
Yeah, actually, I wish to add more slide but based on the guideline you mentioned,
Yeah.
just a picture from the paper, I try to stay with the pictures from the paper. And so otherwise, chymographs. So during the single molecule assay, we are using the TERF microscopy. In the TERF microscopy, we are able to monitor the movement of the motor proteins or any other activators or anything that can bind to them if they are fluorescently labeled along the microtubules. The movement will be captured in the movies, and then these movies can be transferred into chymographs. In the chymographs, you can the position of the motor protein or their cargo along the microtubule is plotted against the time, and this is what you can see. So the x-axis is the time, is the distance they are traveling, and the y-axis is the time. This is why you are seeing. So you can imagine if they are in the case of this the first chymographs, if they are not moving, so the position is not changing by time, you will see just vertical lines. But if they are moving, you will see some tilted lines that shows the particles are moving on the microtubules.
Cool.
Very
And
cool.
so straight down means it's not moving, and the greater the slope, the
Absolutely.
faster they're moving. Right.
Absolutely.
Yeah, that's just really cool. All right, thank you.
Absolutely. The more the slope is, means that they are faster. So
Yeah.
Thanks.
Yeah.
And okay, whatever we have seen till now, it's coming from the in vitro data. So the main question might be, how about if it's the same thing that's happening inside the cell? Because already it has been shown that BICT2 from the cell-based and the virology study, that BICT2 is the cargo adapter of dynein for HIV-1. So in collaboration with Dr. Ed Campbell in Loyola University, we did a set of cell-based and assay over that we analyzed whether knockdown of the BICT-R1 or Hook3 or other activators is affecting the infectivity or not. Because if it is only BICT2, the knockdown of those other activators should not affect the infectivity. And interestingly, they found in both cases, when in different cell lines that they tried, knockdown of the BICT-R1 can reduce the infectivity by 30% and knockdown of hook-3 can reduce by 36% over here in this case. Another a study is coming from the a proximity ligation assay. In this proximity ligation assay is when the two components are close to each other. A puncta is formed, a fluorescent puncta is formed that you can measure it. It may not be really clear in this case in the image because the punctas are so small, but you will see in the case that we have the enveloped protein of the HIV-1, that's a control, is mutated, so the infection is not actually happening. You don't see any puncta, but after but when you are having the actual virus and you are measuring the proximity between the big TR1, because big TR1 is something that hasn't been shown in the other studies. When you are measuring the proximity of the big TR1 with the HIV-1 capsid, they have noticed huge increase in the formation of the puncta just 1 hour after the infection. Means that they are coming together. They are staying, actually during the infection, they are staying together. It was very interesting data. There are even some other supporting data not included in the paper, but recently has been shown that those showing that they are able to complement each other. If you are knocking down, for example, big T2, that you don't see any infectivity, but you overexpress hook3, the infection getting back to 100%. So they are able to complement each other as well. So that was so indirectly showing that what we have seen in the in vitro, it is something that's happening inside the cell as well. So we said that everything was from the purified HIV core that has the capsid plus many other component inside the capsid. So it was which part of those core is interacting. As I mentioned before, the core stay as a whole in the cytoplasm and won't break up. So the capsid of HIV should be the main target for interaction with the dynein. The capsid of HIV made of a single protein, the whole capsid, known as P24, or it is shown as a capsid. The capsid can be expressed and purified can be expressed in E. coli and purified, and under the conditions such as using the polyionic compounds such as inositol 6-phosphate can shake this cone-shaped one, can polymerize into the cone-shaped one, or under the salt condition can make the long tube, mainly made of the hexamers. The 6 of those cap protein coming together make hexamer, plus some pentamer that help with the closure structure of that. If they are just made of the hexamer, they have this open tube one. So I did the same curve assay in presence of the dynein, dynactin, plus different activating adapters. Some of them are like big TR1 are constitutively active, or big T2 are auto-inhibited. And interestingly, I was able to repeat whatever I have seen before with the full core with the capsid itself. It means that from the core, it is the capsid part that is interacting with the dynein, and again, the presence of any activating adapter that is constitutively active can generate motal events on the microtubules. So the next question, so it is capsid. How about the HIV? How about the dynein? Which part of the dynein is involved to that? Dynein is a huge structure, 1.5 megadalton, but it's mainly made of 2 parts, the motor domain part and the tail part. The tail part also made of the heavy chain and several smaller lighter chain, as you can see over here, that I'm not going through the detail. But I was able to purify separately the tail domain and purified separately the motor domain. And I did the coupleting assay in presence of the tail and the motor domain of the HIV capsid that I assembled in vitro. So as you can see, when I don't have any capsid, almost all these components, the full complex of dynein, motor domain, or the tail domain, stay in the supernatant, and I don't have anything in the pellet. But when I'm adding the capsid, as you can see over here, in the supernatant, the only thing that is left in motor domain, while the whole component of the capsid and the tail domain, you can find them in the palate, means they can copulate with the capsid, while the motor domain stay in the supernatant endon. So we know that it is the tail domain. Another piece of data that we have is coming from
So this is basically an assay where you show that in the presence of caps, when you add capsid, they just absorb these dynein, these proteins.
Absolutely, yeah. And the direct interaction, it is happening.
Okay.
Everything is from the purified protein, but people have done this experiment with the clear lysate of the cell to see if the dynein inside the cell can interact with that, and they show that this interaction happens with the clear lysate, not just with the purified protein. So it is something that inside the cell. And yeah, another piece of result is coming from some evidences, some clues in the paper that was available that this LC8 from the dynein is involved in the interaction with the capsid. To see if this is the case and the LC8 involved, I generated a dynein that I call dynein delta DIC 37. DIC is called as dynein intermediate chain, is this green part that I deleted it from the 137 N terminus. And when I'm deleting this part, both LC8 and TCTX1 will be lost. They won't be available in the final structure, and we will have something like that. And I look at the recruitment of the HIV capsid in presence of the full complex of the dynein and this dynein delta DIC 137 to see if this component of the LCA TCTX1 and the N terminus of the DIC play any role. And interestingly, I found that I have significant reduction in the number of the recruitment of the dynein to the HIV to the microtubule in presence of the complex of the dynein, comparing to when I'm using the dynein delta DIC 137. So in other words, it can say that this specific region of dynein playing a major role in these interactions. We are doing further study at this point to further figure out how this interaction is happening, but it's by itself a big project that's going on. The last figure of this presentation is coming from it's coming from this evidences that we had, that we showed that I already showed you that the HIV-1 can co-localize with a dynein already during the movement. But in the background of this dynein channel, you see many other single molecule of the dynein that are moving and not localized with the HIV-1. But something, as already Scott mentioned it, in these chymographs like this that you can see when they are vertical lines or close to the vertical, they means that they are slow, but when they are coming to be close to the horizontal means they are fast. And those dynein one in the background are way faster, comparing the dynein that are co-localized with the co-localized with the HIV. The other things that you can see, it's very clear, the dyneins, the dynein channels, the dynein localized with the HIV-1 is way brighter than the single dynein. In the other words, as you can see, I can see about 7-time increase when I measure the brightness of the dyneins co-localized with the HIV-1. Means that there is a team of dynein exactly involved in that. But I didn't have any control on the number of bind, how many number can bind, and it was hard to measure it. So I developed a kind of model system. It was based on the encapsulin. Encapsulin is a protein cage available naturally in bacteria. And in the collaboration with Dr. Giesen, Toby Giesen in the University of Michigan, they generated a sort of encapsuline that has about 215 same building block, but the N-terminus of this building block has a spy tag on it, and this spy tag are used to interact them with the molecules, with any other molecules that has a spy catcher. A spy catcher interaction is very efficient reaction, almost to 100% yield. And over here, I use big TR1, but really doesn't matter at this point. The most important thing it has was the big TR1 that was interacting, big TR1 spy catcher that was interacting with this a spy tag on the encapsuline. They had this Alexa dye, and over here I had a control. One of these Alexa dye can be on the capsuline, 100 of Alexa dye, 100 of this, 10 of this Alexa dye. So I was able at the end to generate based on the fluorescent intensity of and the plus number of this Alexa dye that I used, I was able to generate a standard curve. And from this standard curve, at the same time, I had dynein exactly labeled with the same Alexa dye, and then measure the fluorescent intensity of the dyneins that are colocalized with the HIV. And based on that, and from this dynein, from this standard curve, I showed that about 10 of HIV-1 can 10 of the dynein can interact with HIV-1. Also, I showed to you that HIV-1 is way slower comparing to the single molecule of the dynein. So I used the same system, but here big TR1 that are interacting with the that are attached to the encapsuline are not fluorescently labeled, but I use them as a system that can move on the microtubule because big TR1 can interact with the dynein and dynactin. And I define them with a different numbers. So for example, one big TR1 per encapsuline, that means 1 dynein per encapsuline, or 10 dynein if I increase the number of big TR1. And then I measured how the motility, how the velocity of a speed will be affected when I'm increasing the number of the big TR1 or indirectly number of motor protein. As you can see over here, when I'm increasing the number of the big TR1 per encapsuline, very clearly you can see, I can see a huge reduction in the velocity of the encapsuline. That sort of can correlate to what I have seen with HIV-1. Although HIV-1 still is way slower, but we can say, because we have seen a team of dynein is involved, this HIV-1, the team of dynein plays some role in the reduced velocity, in the slow movement of HIV-1 on the microtubules.
So, what these data are seeming to show is basically, if you might get about 10 of these big DR1 to bind dynein to bind the capsid, right? And they would slow down the motion.
Yes, at least we can say they contribute to some extent because, yeah, we have seen way slower one. So that's another mysterious part why they are so much slower. But we can say that when we are having more of the dynein, we have a slower movement. Yeah.
Yeah. So, because it's this capsid is like, you showed in one of your data, I think your figure 2 data, right? It's like a sponge, figure 3 data, I think. It's like a sponge for these proteins, the pelleting assay that you showed.
Sorry, yeah.
I think it's further down where you pelleted
The
it.
co-sedimentation experiment.
Oh, this co-sedimentation experiment, yeah. So you show that these are like a sponge, right? So there is something that controls how many can bind and not, is it? It's fascinating, isn't it?
Yeah, but you know that the HIV-1 is made of exactly the same protein. So the building block are exactly the same. It's the other word. There are many space available, many spot available for binding. So you're right. There are many of them available, and when we are increasing the size of them, then binding site possibly increases too.
Right. That's very fascinating. Thanks, yeah.
Thank you. Yeah, you're welcome. And yeah, that's what we are having from this data. When getting back to the original image that I have over here, we can say that BICT2 is not the only one involved in the motility. How BICT2 is activated inside the cell, it's another big mystery that we are working on. That we believe it might be part of the, not hijacking, we call it hitchhiking. So BICT2 is activated by the dynein, with the activated BICT2 is activated by another cargo, and dynein bind to that BICT2. It is already activated by another cargo, and it is the spot the moment that HIV-1 take advantages and bind directly to dynein and move with that. And but at the end we can say dynein, HIV-1, it is way more opportunistic hijacker than we thought before. And to get to the nucleus, it take advantages of any activators and active dynein that is available inside the cell to get to the nucleus and do the rest of the replication. That I can say is a major finding of this paper.
That was great. Congratulations. This is really cool work. Totally enjoyed the images and how you guys put together stoichiometry of binding and movement and so forth. So now we'd like to switch to the interview part of the podcast if you guys are ready for that.
Yeah, so great. So yeah, we have a few questions for you about how the paper came together, but also like we want to learn more about you as a scientist, right? So sort of about your journey, what inspires you. And so Francis and I will take turns asking you questions. I'll ask it specifically to each of, to individuals, so you'll know who to answer and who will answer. And yeah, so I'll have the first question and then we'll take turns. Okay, so Somaye. You did the work. A lot of times with a paper or a study, there's like one moment where you're like, now I know I'm onto something. What was that with this paper?
And that's a great question, actually. Um, as I said to you, I can say, like many other projects, the downtime for this experiment, for this project, is way more than the actual those moment that or exciting moment that I had. I was 2 years. I was checking why I'm not able to see any motile events with the BICT2 that has been already reported in the paper. One of the interesting, it wasn't the aha moment, but it was surprising for me that because I was working with for another experiment, I expressed BICTR1, and I said BICTR1 in contrast to BICT2, it is not also inhibited; it is constitutively active. I just, as a control, I added to my experiments, and to my surprise, I was able to see motile events I wasn't able to see with the BICT2. So I can say that's another one. But it wasn't the only step. I remember I presented this data to one of our microtubule supergroup meetings, and Morgan DeSantis at that moment mentioned to me that you had to have another control. For example, use dynein to see another activator of activating adapter of dynein to see if you can see any motile events or not. And I really didn't expect it, but I got it from them. They had expressed dynein. I got it and added to the experiment. And again, I was able to see that one. And the motile events. I then, to my surprise, because I was checking which part of the BICTR1, because we were thinking that the BICTR1 is involved as a cargo adapter. So the cargo binding domain of that is interacting with the capsid. So I generated truncated version of BICTR1. And again, I was like, I'm still seeing motility. So it was very crazy. And for a while, I didn't have any idea till I ran that drop-off experiment. And so it was kind of opening of the using situation.
Luck favors the brave. You've really
Mm.
been very brave with this project. I really loved how you guys tailored and cut down the proteins to map the domains and stuff. And this capsid story is very fascinating, right? So, Mike, it brings me to the question that I wanted to ask you, okay? So you are like a microtubule aficionado, if I may say that, right? And what do you think? You think that the HIV the virus is teaching you a lot more about microtubule biology? and how do you feel about this adventure that you've taken here?
Yeah, I mean, I guess like for me, the what I want, I think the long-term thing I want to study is how cargoes determine when they get trafficked in the cell. And so HIV, I think, is a nice intermediate where it's a viral cargo. And so that's where it fit in, where it was like, well, if this is hijacking our motor proteins, and we should figure out how it's doing it, because usually these viruses are doing things that other things do in the cell. They're sort of co-opting existing systems. And so part of what I've been thinking about with Samaya's work is like if HIV binds dynein directly for trafficking, there must be other cargoes that do the same,
Right.
perhaps. And maybe native cargoes do. We don't know what the number of cargoes that have been reconstituted with dynein are like very few. This is like the second one. There was a
I see.
Rab6 vesicle, and I guess there's even RNA, but very few. And so I think we're still learning. And so my guess, though, is that there will be others that bind directly. And that gives you this ability to move with any active complex. So for me, that's where it fits in, and it's been fun to learn the whole field of HIV and sort of pick it up. And Samaya and I both benefited from these collaborative grants, pilot grants to really get us off the ground to get us to where we are today. As we still feel like newbies in HIV land, or I do at least. But it's fun to be here.
Yeah, that's cool.
Right. So Mike, I have another question for you. So you've been in the field a while. You're doing biochemistry now. You've done cryo-EM. A lot of times, there's like one experiment or a single image or something that just is like it just burned into your memory. It's like, oh my gosh, that's the gee whiz moment. Do you have anything like that?
Uh,
Yeah, I think for me, or sorry, go ahead, Samaya.
no, that's good.
You
Thank
should
you.
both answer. So the first,
Mhm.
Mike, you do it, and then
No,
Samaya, then you do it.
yeah,
Sure.
I already answered this.
Okay.
Okay. For me, it's not the HIV project. It's more of my PhD work with Eva Nogales. It was this cryo-EM, this large transcription factor complex. Before the high resolution, we still could see blobs of it changing shape. And for the PhD, they're like, here's DNA. This thing binds DNA, get the structure. And we essentially were stuck for like 4 years. And we could see it in the images, but couldn't get reconstructions of it. And it was redoing like the, umpteenth control experiment, tilting the samples. And I was at this Gordon conference for cryo-EM, reanalyzing this like tilted data when I realized that my protein like changed shape. And the reason why nothing worked is that it essentially was isomerizing. And then at low resolution, you wouldn't tell, but once you can see the shape of it, you could tell this domain was bouncing back and forth. And that was like this huge breakthrough of like, oh, that's why it wasn't working. And for me, the exciting thing was I could go through all the data I ever had collected, and it was always there. And so this shape was always changing. I just didn't appreciate it. I even found data from before when I was in the lab, and it was already there. So it was that stands out as like satisfying because it explained, 5 years of experiments, and it was always there.
That's right.
Cool.
How about you, Samaya? What is the image that just has frozen in your mind that inspires you to continue this in your scientific journey?
Um, I can say the moment that the first figure of this paper by itself is a little story, but the moment that I saw that dynein is enough by itself to bring was really, it was like meet so many pieces of puzzle together, and then suddenly all of them came together, that was something like that. Yeah.
So, I want to dig a little bit deeper into, so when did you start using microscopy in your scientific journey during your PhD
Just
and
my,
postdoc?
yeah, it is a start at both cryo-electron microscopy and turf microscopy was started in Mike's lab. Before that, I was using it, but for example, I had a little bit of the cryo experience before working with but it was very limited. So I can say the main thought of microscopy in my journey was in Mike's lab. Yeah.
I
Before
see.
that, yeah, my background is mostly on the protein modeling and protein engineering.
So it fits directly very well into your scientific career image, I guess, right? Do you
Yeah,
like
yeah.
microscopy?
Yeah, absolutely. Yeah, both of
Okay.
them, both side of the cryo-electron microscopy and also this fluorescent microscopy, single molecule assays. All of them is my
Can I
interest.
also ask you this other question because we are all microscopists here, and or when we take an image, we are like so excited. It's like, oh wow, look at this image, man, it's so beautiful. It means some. And then you say, oh, we have to start quantifying and putting number to it, and then you start just getting crunched. How did you feel? How was your quantifying journey from the
Um,
image?
you mean that quantifying the
Figuring
macroscopic?
out how to quantify your data, coming to the chymographs, and plotting all those things. How did that journey go?
Yeah, we were very privileged of having this microtubule group meetings, super
Yes.
group meeting, and another super group meetings that is going on with the other universities. And from the very first week that I was in University of Michigan, because we have a great faculties working on the microtubules, we had this super group meeting, and I was I was very closely introduced at the very beginning of my journey to how to quantify them and how important it is quantifying and how studying way more detail that what we are seeing on what we are seeing those quantification more detail those quantification can provide for what we are studying. So, for example, this is just we are seeing one single motor moving on the microtubules, but people are looking at the dual time, I don't know, velocity, run length, many parameters that each one by itself can provide a new data and to
Information.
differentiate the proteins from each other, motor proteins, different mutation and everything. Yeah.
So,
Yeah,
so these big groups and these discussions actually very helpful to say, oh, maybe we should quantify
absolutely.
these things.
Yeah, yes, absolutely. The great part of working on the microtubule in University of Michigan is being involved in this super
How
group.
was your experience, Mike? When you start your PhD and you're like just being introduced to quantification on the computer of these images, right?
Hmm.
From
Yeah,
there
same. I mean,
to where we are
yeah,
right now.
yeah, I know. I mean, I think for me, it wasn't fluorescence microscopy yet. It was like electron microscopy. But
Electron
yeah, the
microscopy.
first time you look at a protein in negative stain EM, it's amazing. Like you're just looking at a protein. It's that was it for me. Just
Yeah.
I'm a visual person. I didn't really realize at the time, but I seeing it was great. And then when I learned about turf microscopy, that's when I was like, oh, if I do a postdoc in microtubules, that's a great assay. It's single molecule, but it's also like you get a number of events per movie, so it's kind of nice. And so it felt like it worked really well with cryo-EM in terms of amounts of things. And so sometimes it feels like we're very basic because we just look at stuff. We look at it in electrons, we look at it with lasers. That's it.
Yes.
We don't do anything like indirect and fancy. It's just we
Right.
just see it.
I have. Yeah.
All right.
So
So one last question will be for both of you, and then we'll move on. So uh, I'll start with Samaya. So who is your scientific hero and why?
Yeah, I was thinking about that question a bit, and I think from many years ago that I was undergrad, I was really uh, I was fascinated and interested in the life of Barbara McIntosh.
Hmm.
So I read about her and I studied, and it happens that few years I went to the Cold Spring Harbor laboratory and I visited her lab and everything. And so still, I think that she is my hero in the field of
Mm.
science. Yeah.
That's great. Mike, how about
Mm.
you?
I think people who can take sort of like biochemical observations and just take it all the way to the end up to humans and health. And so I think for me, people who stand out would be like Ron Vale, who could go from discovering a kinesin to structures of kinesins to functions and all the way up into organisms and humans. And so I think to me, he stands out.
Yeah, yeah. Great. Both are great answers.
all
All right.
right.
So Ash, you want to finish us up?
Yes, Mike and Samaya, thank you so much for joining us today. And it was so nice to hear your wonderful paper, the results from that, directly coming from you guys. And, really appreciate it a lot. And thank you for joining us.
Mhm.
Thank
All
you.
right. So that does it
Thank
for
you.
this episode. We hope you all join us again for the next episode of Structure Club.